Hello, Dr. Clark here, and in this video I'm going to give you a brief overview of the seed germination experiment. For this experiment, what you're going to do is examine how light impacts germination frequencies for two different plant species, lettuce plants and lentils. And what you'll need to do is you're going to prepare a set of petri dishes, very much like this dish here, and you're going to put seeds of a given species into the dish. In this case is actually some pea plants. And then you're going to add some water. You're going to seal this dish closed with some parafilm, and then you're going to either wrap the dish in aluminum foil to keep it in complete darkness, or you'll leave the dish exposed to the light. And for this project, it's really important that your seeds that are to be kept in the dark experience next to no light as you prepare them. So if you're able to do so, one option will be to use the SciWriting lab space to prepare your seed dishes. And then there's a green safe light that may either be somewhere in the SciWriting lab, or else Dr. Helm, our resident plant physiologist, might have the green safe light in use somewhere for plant physiology. So you can go and ask him where to track down that safe light. And you can use the safe light to then prepare your in-the-dark seeds. You'll want to then check on your seeds that are in the light at regular intervals and examine them for signs of germination. So in these pea seeds, it's pretty obvious here. You can see, if I hold that up, there is a white radical sticking out of this pea seed. So this pea is very much germinated. So that's the same type of sign you'll see for your seeds. And once you've reached a point where you're observing a reasonable amount of germination in your light germinated seeds, so in my dish, almost all of these pea seeds are germinated. Then you're going to want to take the foil off of your dark germinated seeds and count the number of germinated versus non-germinated seeds in your dark germinated treatment dishes. And then as a final step, if you do indeed observe lower germination frequencies in your dark germinated seed dishes, you should then leave those dark germinated seeds exposed to the light for the same time interval as for your light germinated seeds, and then come back and repeat a final count of how many seeds have subsequently germinated. And this last step is important because it's going to help you determine whether the outcome that you observe has something to do with the viability of the seeds you're working with, so maybe they're dead, or whether there's something else going on with the seeds that are kept in the dark. So now, it's important with this project that you check in with your teammates partway through, both to help keep yourself on track and to see, for example, whether you are observing similar or different outcomes and why that might be the case. And once you've completed the pilot experiment, you should have a good set of ideas to explore and take this project further for your main experiments, where you'll have an opportunity to investigate in much greater detail just how it is that seeds are detecting and integrating information from the environment when making the decision that it's the right time to germinate.